A good day to everyone. First, I'd like to thank the organizers for inviting me to this event. Today, I'm going to tell you the story of our research during the last years, elucidating the biological roles of high molecular weight immunophilins, using the glucocorticoid receptor as a model since they were first discovered associated to steroid receptors. Glucocorticoids regulate physiological homeostasis in every organ of the body. There is no tissue in which glucocorticoids do not have an effect. Glucocorticoids were named as such due to their ability to promote liver gluconeogenesis and to decrease glucose uptake in muscle and adipose tissue. Therefore, glucocorticoids enhance hyperglycemia and insulin resistance, among other actions such as immune response, apoptosis, reproduction, differentiation, etc. Glucocorticoids bind glucocorticoid receptor, a ligand-activated transcription factor belonging to the nuclear receptor superfamily, whose structural domains are depicted on the top. GR affects gene expression by both mechanisms, transactivation and transrepression. For years, it was believed that the presence of HSP90 bound to GR was essential for its cytoplasmic localization. Steroid binding dissociates HSP90, a phenomenon named transformation, which permits the free diffusion of GR towards the nucleus. Soluble GR exists as an oligomeric complex of chaperones that are required for GR to bind the steroid, such that new GR does not bind hormone and is targeted to proteasomal degradation. HSP90 is the hub of the complex, which also includes HSP70, P23, and HOPE, P60. HOPE is required during the early steps of GR assembly, bringing together HSP90 and HSP70, but it is not present in the final mature heterocomplex because it is replaced by the high molecular weight immunophilin. Both HOPE and immunophilin interact with HSP90 via TPR domains, shown here as a black crescent, that is, sequences of 34 amino acids repeated in tandem. Immunophilins are a subfamily of molecular chaperones that show two main properties. One, a domain with intrinsic enzymatic activity of peptidyl prolylasomerase from here onwards PPIase, that is the cis-trans isomerization of peptide bonds N-terminal to the proline. The other is the binding of immunosuppressive drugs. If they bind the undecapeptide cyclosporin A, they are named cyclophilins or simply CYPs. If they bind the macrolide FK506 or tacrolimus, they are named FKBPs for FK506 binding proteins. These are the best studied HSP90 binding immunophilins. They have the PPIase domain shown in yellow where the drug binds, and in blue the TPR domain through which where HSP90 bind and through which they interact with client proteins such as GR. When these immunophilins were first discovered, their functions were unknown, except for the fact that they were not involved in immunosuppression, a phenomenon mediated by low molecular weight immunophilins only. Therefore, we focus our interest on the elucidation of their function, and these are our early observations. It was known that the nuclear accumulation of GR is complete after 10 or 15 minutes. Sucrose density gradient shows a typical untransformed 9s peak that shifts to 5s peak upon the steroid binding. This is due to GR transformation. Interestingly, we found that 10 minutes after the addition of steroids, when the GR is becoming fully nuclear, almost 70% of GR still shows in the 9S form. It means untransformed isoform. Is this GR bound to HSP90? To test this, the 9S nuclear form of steroid-treated cells were pulled, incubated with an anti-HSP90 IgM, and the gradient was run again. The 9S peak shift to 11.4S. The increase in the molecular mass was equivalent to 1 million. It means the mass of the IgM. So receptor transformation is not an early event. In short, HSP90 is still associated to GR, probably to the nuclear fraction of the receptor. This was proved by immunoprecipitation of soluble GR isolated from nuclear extracts, free of cytoplasmic contamination. Note that the HSP90 co-immunoprecipitated with GR in the nucleoplasm disappears after 10 or 15 minutes of steroid binding, whereas the nuclear pool begins to be attached to the pellet, it means to chromatin. Then we ask, is FKBP52 required for GR nuclear accumulation? L929 fibroblasts were microinjected with antibodies against the PPIase domain of the immunophilin, against the hinge region of the immunophilin, or against HSP90. The control is a non-immune IgG shown here in panel A as NI. Microinjected cells can be distinguished in green due to the co-injection of fluorescent dextran. In contrast to cells injected with the non-immune antibody in panel B or the anti-hinge antibody in panel F that shows GR entirely nuclear in the presence of steroids, cells microinjected with the anti-PPIase domain, panel D, or anti-HSP90, panel H, still shows GR in the cytoplasm. Therefore, the HSP90 FKBP52 complex should play a role in GR retotransport. FKBP52 shows high homology and structural conformation with the immunophilin FKBP51. They share about 75% amino acid similarity. This led to investigate the presence of 51 in the GR complexes and how this may affect GR subcellular localization. Fibroblasts were placed on ice and stimulated with a steroid. This permits steroid binding to GR, but not its translocation, which is an energy and temperature dependent phenomenon. GR was immunoprecipitated. Clearly, the immunophilin FKBP51 is released from the complex upon the steroid binding. At the same time, FKBP52 is recruited as well as binding. This makes sense because the receptor should move towards the nucleus. NIH3T3 cells were transfected with GFP-GR. GR is mostly cytoplasmic in the absence of a steroid, and after 20 minutes with a steroid, it becomes entirely nuclear. Here we see the nuclear translocation rate. The HSP90 inhibitor, galdanamycin, should prevent GR movement, and cells should look like this one. However, the receptor, when galdanamycin is inhibiting the heterocomplex, is still moving, although it moves slowly. It takes almost 1 hour to reach the nuclear compartment. Is this due to the toxic effect of the drug? To answer this, we use a non-pharmacologic approach, the overexpression of the PPIase domain, which should compete with dyne binding. The results were quite similar, as well as when we overexpress the TPR domain, which competes with the binding with 52, not shown here, or when we use an inhibitor of the ATPase activity of dyne. All the translocation rates were similar. This is telling us that there is an active dyne-dependent mechanism which is efficient and brings the receptor to the nucleus in 10 minutes. It's rapid, as well as a passive and less efficient mechanism, probably diffusion. The HSP90 FKBP52 complex is the linker between the motor protein and the cargo, in this case GR. All immunophilins found in a GR complex bind dyne, FKBP52, CYP40, and PP5. All of them co-immunoprecipitate with dyne, except FKBP51. This explains why 51 must be released upon stereobinding. GR must move towards the nucleus. Because HSP90 is a dimer, we ask how many of these redundant immunophilins are simultaneously bound to cytosolic complex, 1 or 2. Purified HSP90 was mixed on ice with purified FKBP52, and complexes were cross-linked with glutaraldehyde. Both HSP90 binds, monomer and dimer, fade when FKBP52 is present in favor of this band of equal to 240 kilodaltons. Nothing is shown above. This argues in favor of a stoichiometry of 1 TPR protein per HSP90 dimer. GR nuclear translocation was then analyzed in FKBP52 knockout cells. Note that GFPGR in green is still cytoplasmic in the presence of a steroid in cells where FKBP52 expression is abolished, whereas it is entirely nuclear in the red cells that represent FKBP52 reintroduced by transfection. It is not due to failure in steroid binding as it is shown here. The nuclear translocation rate of the receptor is slower in knockout cells compared to wild-type cells or the same knockout cells where FKBP52 was reintroduced by transfection. Also, note that the nuclear localization, primary nuclear localization of GR in knockout cells is lower than in wild-type cells, suggesting that the immunophilin could anchor to GR to nuclear structures. Is the heterocomplex disassembled when GR reaches the nuclear pore? To answer this, GR was produced in SF9 cells infected with baculovirus. Cross-linked the endogenous complex with DSB, and this cross-linked complex was offered to E82 cells, which are GR negative knockout cells, permeabilized with digitonin. After the incorporation of the proteins into the cells, digitonin was washed out and cells were stimulated with dexamethasone. It can be seen that the green fluorescence of GR by indirect immunofluorescence is entirely nuclear. This complex is cross-linked. There is no possibility of transformation, implying that GR transformation is indeed a nuclear event. This was proven 2 years ago by 2 independent labs using different biophysical methodologies. Interestingly, confocal microscopy showed that nuclear GR co-localized with nuclear matrix proteins, such as NUMA in green, and also with FKBP52. These magnified images show the nuclear speckles of NUMA with GR, yellow, GR with 52, magenta, 52 with NUMA, yellow, and all three together in white speckles. The biochemical purification of the nucleoskeleton confirmed the co-purification of 52 and GR as shown by the markers, matrix markers NUMA and lamin B, and the immunoprecipitation of NUMA from nuclear extras pulled down FKBP52, right? In this case, cells were permeabilized with tritone that makes more visible the presence of nuclear speckles. If this permeabilization is followed by a treatment with high ionic exchange to disassemble nucleosomes and chromatin is degraded by DNase, you still can see the true localization of FKBP52, GR, and NUMA in nuclear speckles. Immunophilins are also regulators of GR transcriptional activity. 51 is a strong inhibitor, which is another reason for this immunophilin to be exchanged by 52 upon steroid binding. However, the overexpression of 52 does not shows just a mild stimulatory action on transfection. Nonetheless, in 52 knockout cells, this is more evident. In knockout cells, there is a shift to the right of the concentration curve response compared to wild type or knockout cells where 52 was reintroduced by transfection. Our interpretation is that this mild action in normal cells of 52 is due to the influence of the endogenous 52, whose expression level is enough for a positive effect. Note that both immunophilins are antagonistic. The negative effect of 51, shown here, is reversed by expressing increasing concentrations of 52. In short, a novel mechanism of action for a steroid receptor is proposed. The upper part shows the classic mechanism posits during the 80s. In the absence of the steroid, we propose that GR-HSP90 complex contains FKBP51, a protein that regulates negatively all steps of GR pathway, including its nuclear anchorage, as we have recently published. When the steroid binds, GR changes the conformation, exposing the hidden nuclear localization signal depicted here in pink color, and also exchanges 51 by 52, which in turn recruits dyne. The whole heterocomplex passes through the nuclear pore where transformation takes place. GR dimerizes and recognizes hormone responses elements, triggering transcription of target genes. Curiously, one of these genes is FKBP5, which encodes for FKBP51. Therefore, GR induces the expression of its own negative regulator to feedback its own action. This yin-yang of immunophilins reflects the GR-dependent action and explains the pleiotropic effects of GR, sometimes antagonistic. For example, glycogen storage in hepatocytes and glycogenolysis in lymphocytes, or cell division in uterus and apoptosis in thymus. The relative expression of 52 and 51 is different in these systems, and consequently, it is so the composition of the GR complex. Thus, the 51 to 52 relative ratio of expression regulates the nature and intensity of the final biological response. A similar explanation may apply to actions of other factors such as NF-kappa B. Recent papers, we demonstrated that NF-kappa B is similarly regulated by immunophilins as GR is. At first, these findings related to retro-transport of GR and the 51-52 yin-yang were resisted by many people. It makes sense because the new model tears down a 35-year-old unproven dogma, but an ingrained and heuristic dogma at the end. When these observations were reproduced, the same people began to accept this model, which is also being applied to a number of soluble proteins, some of which are listed here. Special thanks are due to this family who has made this project possible. And thank you all for your kind attention. I'll be happy to answer any question you may have. Thank you.